Hello lovers, in this video we're going to be looking at genetic variation for your A-level biology. Now please make sure you get all of the examples that we're going through here and that you are able to apply them in the exam, because these are the sort of skills that the examiners are looking for to make sure that, you know, we are confident with what we are doing. On the website, Dr. Edwards has written you a set of questions specifically to go with this video.
Genetic variation. So our definition of variation hasn't changed. It's the difference between individuals in a population. So obviously this is talking about individuals within the same species. So really we're talking about this idea that how genetically, in terms of alleles and other things like that, but also just how are they different? And that's what we're going to look at. So there's two causes of variation, and we need to think about how genetic variation is created as well. So there are different causes of variation. There's environmental causes. These can include climate change. So that means kind of drastic changes in abiotic conditions, the availability of food or water in the habitat where they live, the intensity of light and soil nutrients. These last two are pretty much going to affect plants only. These are going to affect how organisms grow, because obviously it's going to be dependent on how much nutrients they're going to get, as well as how much they're going to be exposed to various different temperature and weather effects. And these can be what lead to causing adaptations to organisms over time. The other type and cause of variation is genetic. Most organisms vary between each other because they have different combinations of alleles. So let's think brown hair versus blonde hair versus ginger hair versus different eye color, those types of things. So all humans have those same genes, but their alleles are what makes the variation between them different. Mutations can cause random changes to the DNA sequence or the structure and number of chromosomes. And we've talked about how that can then have a knock-on effect on proteins, which is what usually lead to the various characteristics that an organism has. Meiosis, we've just talked about. So we know that there is a couple of ways that meiosis can increase genetic variation through independent assortment or segregation and crossing over, which creates different combinations of alleles. Also, the fertilization of gametes, so the act of sexual reproduction itself, because it's random which two male and female gametes actually fuse, creating the new parental combinations of alleles in that gamete. So it's always random every single time which sperm meets which egg in the case of humans. And therefore, we've got this another way of including and increasing that variation in genes. So one of those causes of genetic variation is chromosome mutations. And we need to be able to know and recognize some of the specific types of chromosome mutation. These can occur during meiosis. So the first one is deletion. This is where part of a chromosome and its genes, so all the genes that are in that section of the chromosome, are lost. So you can see here my chromosome is much shorter than the original. Then we've got inversion. This is where a section of a chromosome will break off, detach, go through a 180-degree kind of twist, flip, and then reattach. So in this case, you can see obviously that large band on the original chromosome is now underneath. So it's lower down because that section of those kind of four bands has kind of broken off, twisted around, and then reattached. Although we've not lost any of these genes in this type of chromosome mutation, they might now be in the wrong place. So obviously at the start of each gene, there's the regulatory sequences, which are going to determine how it's expressed and when it's expressed. And those may have been interrupted and disrupted by this moving around, and now they might be in the wrong order. So the DNA is obviously linear in chromosomes. It's read in one direction, especially when we're talking about transcription. So we need to make sure that sequences that control those genes are in front of them, and that might not happen anymore. Duplication, this is easy. A section of a chromosome may be duplicated. So I've got an extra kind of few bars on the end of this chromosome, which is now an extra little bit longer. And this is just the same genes in that section will have been replicated again and sort of tagged on the end. So this may sometimes lead to overexpression of genes, because if they get expressed twice because they're both there, then that might be harmful because we're going to make too much protein, for example, and then there'll be more protein than we need, and that could interfere and disrupt various metabolic processes. Translocation. So this is obviously very similar to crossing over, except this is not being done between homologous chromosomes. So a piece of one chromosome is going to break off and then attach to another chromosome. Where it breaks also might be an issue. Again, if it breaks in the middle of a regulatory sequence, or if it breaks in the middle of a gene, cuts into a new gene when it reattaches, that can obviously be a problem, because then we can have kind of complete nonsensical bit of sequence. Yes, so this is kind of similar to crossing over, but not where the same genes and loci on the same chromosome, so not between homologous chromosomes. And then finally, we've got non-disjunction. So this is where a pair of homologous chromosomes or chromatids will fail to separate during meiosis. This means that one gamete will have an extra chromosome. So instead of just receiving half of a pair of homologous chromosomes or one chromatid, they get the whole pair of homologous chromosomes, or they get two chromatids. And so this means that we're going to have an extra chromosome where we shouldn't. Instead of a pair, we're going to have a triplet. So after fertilization, the embryo is also going to have one extra. So this is the case for Down syndrome, which is caused by what we call trisomy 21, where we have 3 copies of chromosome 21, which you can see here. Now, there are a couple of terms that we use to describe having extra chromosomes. One of them is aneuploidy, which is where the chromosome number is not an exact multiple of the haploid number, which is what would be an example here caused by non-disjunction. And then we have the other term, which is polyploidy. This is when there are more than 2 copies of each chromosome. This can happen when a diploid gamete and a haploid gamete fuse. So a triploid organism is produced, as an example, because this time they will have 3 copies of each chromosome. So in aneuploidy, the chromosome number is not an exact multiple, and trisomy 21 would be an example because we've just got 3 copies of just one chromosome. Whereas in polyploidy, we're saying we would have 3 copies of every single chromosome from 1 to 23, sorry. So this is sort of the difference between those two terms. Often polyploidy is seen in plants, specifically cultivated plants or plants that we have kind of selectively bred, because when we've been mixing sort of pollen and eggs and sort of fertilizing different plants, we might have mixed plant species or varieties that have multiple different numbers of chromosomes. We also need to be able to explain in detail how meiosis increases genetic variation at the various stages. So firstly, crossing over happening in prophase 1. The reason it increases genetic variation is because it creates new combinations of alleles that wouldn't normally be found on the same chromosome, and then these are inherited together. So let's have a look. This is my homologous pair of chromosomes. For gene A, I have 2 different alleles, capital A and little a. And for gene B, I have 2 different alleles, capital B and little b. Now, the capital letters would normally be inherited together on that chromosome from parent 1, and the lowercase letters would normally be inherited on the pink chromosome from parent 2. And that would be the standard and normal way that those alleles would be inherited and put into gametes. So we normally say that that's about 48% of the time they would end up as they are like that in the gamete. But the reason this doesn't add up to 100 is not 100% of the time are they inherited that way, because crossing over could have occurred between these 2 chromosomes. So if that's the case and we swap a top section of the chromosome with each one, so the lowercase a ends up on the green chromosome and the capital A ends up on the pink chromosome, then what we've got is a different combination of those alleles that can then be inherited on that same chromosome. So that's about 2% of the time would that end up in the gametes. We call these recombinant chromosomes, and the ones that are the same as the parents, the parental chromosomes. So this is something that obviously increases genetic variation. It doesn't happen particularly very often necessarily. It can be quite rare, although crossing over happens a lot, but it doesn't necessarily always mean you get a completely different combination. It depends where the crossing over occurs. Independent assortment in metaphase 1. This is where each of those 23 pairs is randomly arranged either side of the equator when they line up. So the maternal and the paternal chromosome in each pair goes either side of that equator line, and there's nothing to control how that happens. It's just random. This means that those chromosomes are independently assorted into gametes. So there's no link between them. They're independently taken at different sides into the gametes to make new combinations of maternal and paternal chromosomes. This is why you are only a certain amount related to your siblings, because you all share chromosomes from both parents, but you all have a different random combination of chromosomes from each parent. So therefore, that's how you end up having similar traits that you can share, but sometimes you'll have traits more in common with one parent than another. If we work this out, we actually can work out that there's 23 pairs of chromosomes. So 2 to the power of 23 is how we can calculate the variety of each gamete we would get. So all of those different combinations of chromosomes that could possibly happen means there are potentially trillions of combinations if you think about the fact that that's for each gamete. So 2 to the power 23 is for the sperm, and then 2 to the power 23 for the egg, and then they combine together randomly again when they fuse. So we get a lot of variation through independent assortment.
Ouch! This is why in some videos I'll explain scratches.